I too would like to welcome everybody to the seminar series for this fall. My name is David Mokler. I'm the chairman of the Department of Biomedical Sciences here in the College of Osteopathic Medicine. I'd like to introduce our speaker today is Dr. Megan May. She's been with us now for 4 years?
4 or 5.
5 years, something like that. She's a very, very dynamic and interesting person in terms of the research that she does. She does a lot of different work on emerging diseases, and obviously with the all of the information out there about the Zika virus. I was down in Georgia just a couple of days ago to see the eclipse, and they've got huge billboards all over the place about the Zika virus, warning about the implications. In the airports, you can see it there. So it's really on everybody's mind about the disease pattern here. And Dr. May has been doing a lot of work along those lines. So I'm going to introduce her and let her take off.
All right. I'm glad to see some of you guys that I saw in class this morning decided to hang out a little while longer and tolerate me. I'm going to turn these lights down a bit because there's some fluorescence microscopy images in here that might be a little easier to see if the lighting's low. Okay. I'm really delighted to give this talk today for a couple of reasons. I've given several CME talks about Zika for clinicians trying to go through the latest case reports and some drug repurposing and basically trying to get a handle on what we can do for these patients. And I've given a couple of quick talks on some of the basic research that we've done in my laboratory, but I have not yet given one that's both. So here we go. This could be interesting. All right, so the first thing I want to point out is that I have nothing to disclose. That's actually not true. I have plenty of interesting things to disclose, just none of them are relevant to this talk. It's a conversation for Friday evening sometime. Anyway, so I'm going to give you an outline of how this is going to go, because this is this odd kind of hybrid between basic research and CME. So I'm going to give a review of the Zika virus epidemic in the Americas. And this is going to be more extensive than I normally would give in a research seminar. So for those of you who are saying, oh, get to the data, come on, man. This is going to be more extensive, but it is coming. So that's a little more on the CME side. Then I'm going to talk some about the genetic diversity of some of these emergent lineages of Zika virus that we've already published a paper on. That's more on the research seminar side. Then we're going to talk for most of this time about some of our cell binding studies and what that has to do with Zika's emerging pathology. So that really fits under both categories. And then I'm going to talk real briefly about some of our future directions, but also some of the future directions clinically for Zika, and that really goes under both as well. All right, here we go. So Zika virus disease historically was something that absolutely nobody cared very much about. And that was because if you look at the disease that it caused, you had sort of this mild, weakish lung fever, maybe a headache. Some patients would see this as a viral exanthema. You'd have this maculopapular rash that never ulcerated, never really did anything interesting. You might see some joint pain. In some patients, you can see conjunctivitis as well, but this is not a lethal illness. This is not something that people even got complex infections out of. It was a junky arboviral fever that no one paid attention to. Of course, now we see this virus a lot differently. The recent emergence of Zika virus in the Americas, in the Western Hemisphere, really alerted us to the fact that there was a new and emerging pathology that this virus was causing. And this is for a few reasons. A lot of it had to do with the population density of Brazil. We had an incredible number of new clinical cases, and Brazil has a has a pretty good and intact healthcare system. So these patients were able to be diagnosed well and monitored, and it really gave anybody interested in public health and epidemiology an inkling that something very important was going on with this. It showed us that there was an emerging pathology here that no one had seen before, and that was that this virus now seemed to have a neurotropism, so it could pass the blood-brain barrier, and it could cause neurological disease, and also that it had a placental tropism, meaning it could cross the placenta and infect the fetus. Okay. And so the fetuses that tend to be born of Zika-infected mothers, what was noticed at first was that they had this very profound microcephaly. So you can see in that image, you have a microcephalic infant. Their facial features are completely normal in dimension, and yet the back of their skull is very, very small and truncated. And these babies have tremendous health implications from this that we'll talk about in a minute. We also learned that that's not the end of the story for babies born to Zika-infected mothers. Okay, so Zika virus disease in the Americas and why this is different and why we now care very much about this disease, whereas we didn't before. In order to appreciate it, you have to kind of look at the history of this disease. So this is, we talk about it as this new thing, and it's terribly scary, and it's on the news all the time, but this is not a new virus. This virus was first discovered in 1947 on the African continent. So for the first 60 years of its existence, it was just this junky fever no one cared about. And one of the things that's important to note, it's difficult to see, but there's actually a map of the world in very faint colors back there. When it was in Central and West Africa, you had fevers. It left West Africa and started going on to the Asian continent and then into Southeast Asia, and you still were seeing it as this kind of transient fever, even when it got to Indonesia. In around 2007, people started seeing this differently. So this is an article that came out in New England Journal of Medicine in 2009 describing an outbreak of Zika on the French Polynesian island of Yap. And this was suddenly a disease that was very aggressive. You found that enormous proportions of the population were infected, whereas in Africa, it was around 5% of the population. It was not particularly aggressive or invasive. In addition to that, we would start to see in Southeast Asia the very first inklings that maybe this is doing something to the central nervous system, but nobody had really pieced it together very well. There is no evidence that it did that on the African continent. Okay, so just to give a quick view of the outbreak history and where this came from. This is a maximum likelihood phylogenetic tree that my colleague and I generated using whole genomes from several Zika virus strains. And the way that you read these in a nutshell, I'm going to see if I can point at this because most of us are on this side of the room, is the shorter these lines are, the more closely related those strains are. The longer they are, the more distantly related. So everybody down here, with one exception, is a strain from either Central America, South America, or the Caribbean. Everybody up here is from Southeast Asia, and everybody way off on their own lineage over here, these are all African strains. The exception is very important for looking at the outbreak history here. This strain right here, HPF 2013, this is from French Polynesia. And you can see that this guy fits down here. This strain is thought to be the progenitor of the one that jumped over into the Western Hemisphere. The interesting thing about this, you can tell from these guys being so very closely related to each other that this was, in all likelihood, one patient who went from Southeast Asia to South America and then seeded the entire Western Hemisphere. That's pretty amazing when you think about it. But in any event, that seems to be what happened. Clinically speaking, when we look at Zika virus, again, classically it's mild febrile illness. You may have conjunctivitis, which is a little noteworthy with regard to other viral exanthemas. The other thing that's worth pointing out here is that you have an itchy rash. That's a little different. And it turns out the itchiness of this rash is what clued the clinicians in Brazil into the fact that they were dealing with a different virus. There are a lot of viruses that circulate in South America and Central America that fit this clinical presentation exactly. You have dengue, you have chikungunya, you have mild cases of yellow fever, you have West Nile, you have all kinds of viruses that this could be. And normally, when patients go in, physician sees them and says, yeah, you have dengue, go lay down take some fever control medicine, you'll be fine in a week or so. It was the fact that they were seeing so many patients come in all at once, and that they were saying, but my rash is itchy, it's driving me crazy. In that case, you had some very, very astute clinicians in Brazil who then contacted the World Health Organization and said, we need to do some sequencing, some molecular biology diagnostics, because we think we have something different here. So kudos to them, and let that be a lesson to you as future physicians. It's not, okay, and I realize we just talked in class this morning about empirical diagnosis and how you usually think of the most probable thing, but every so often, I think outside the box. Sorry for the irony of this juxtaposition. Anyway, in the emerging presentation of Zika virus disease, again, you see, in addition to this, you see a neurotropism. In adults, this can present as Guillain-Barré syndrome. It can present as encephalitis or meningoencephalitis. And then with regard to placental tropism, you now see something we're calling congenital Zika syndrome, or CZS. That does not flow very well. The most severe presentation of this is that you wind up with microcephalic infants. And so here's another, this is a very famous picture that we saw a lot in 2016 on the news. This is not the end of the story for these infants, though. This is just the most severe presentation. The first inkling of that came from this paper that came out in October of 2016, so not even a year ago. This was looking at microcephalic and non-microcephalic infants. So what you're seeing here is brain imaging of infants born to Zika-infected mothers. Importantly, all these ones circled in red, these were infants that had head circumferences within normal limits. At the time, prior to this publication coming out, what we were advising physicians to do was to monitor infected women by ultrasound and look at the head circumference of the developing fetus. If it was within normal limits, the advice that was given to these pregnant women was, your baby is okay. And then when these studies came out, looking at the imaging, these babies are clearly not okay. Right, so you have anatomical abnormalities, you have atrophies, you can see calcifications in some of these infants. So this is something that we learned more or less the tragic way, that it's not just microcephaly. There are ongoing other cerebral abnormalities with these babies. So this is a much bigger problem than we thought it was. And just to think clinically about how not only these non-microcephalic babies, but certainly microcephalic babies, are, what challenges they face. They have severe cognitive deficits. They have blindness. They have deafness. They have seizures. They have swallowing difficulties. They have all kinds of gross motor defects. And in addition to this, this is something that socially has been a big challenge in Brazil in particular. These babies are hyper-irritable. They're born with very much the same affects as drug-addicted babies. So they sleep 20 minutes at a time. They scream constantly. They're very irritable, and they're very, very difficult to soothe. So this puts an incredible stress on these families. And they're often, if you have families with multiple children, it's incredibly difficult. And so we're having a lot of, in Brazil, the term has emerged, Zika widows. So you have a lot of kind of instant divorces happening, where you have now women that are left with these highly disabled children in addition to their other children. So this has put a real strain on the social infrastructure. And that's something that we need to keep thinking about from an interprofessional education standpoint, right? Because this is a clinical issue that has major social impacts. Okay, so I'm going to go through the rest of this a little more quickly because that was, I think, the juiciest clinical tidbit that I wanted to get across. So I mentioned that one imaging study that was really the first one, but there have since been multiple imaging studies of infants born to Zika-infected women. There are additional and more specific lesions that we can see now. So you can see there's really a big theme here of hypoplasia and atrophy in the brain, so that you have this just arresting of development in these fetuses. And that's quite tragic. Another thing that's come out that's been very helpful is that when you think about these microcephalic infants or infants with brain deficits, even if they've got a normal head size, it makes you think about what are these infants going to face later in life. So we can look at some of their just overt disabilities, but then you have theoretically a whole spectrum of disabilities that we may not be able to predict yet. So you might have an infant who's got motor control, but are they going to have behavioral problems? Are they going to have sensory problems? And so we turn to, on the basic research side of things, some animal models to try to get at those questions. So this is from a study that was published in May. And what these investigators did was they developed a mouse model of congenital Zika syndrome. And what they found was in the pups that were born to Zika-infected mice, they had severe motor deficiencies. They had vision problems. Their behavioral testing is ongoing, but I actually found they've published some of the raw data on ResearchGate. And it seems that they've got a lot of behavioral abnormalities as well. So they seem to be more prone to anxiety and maladaptive behaviors. So that's not promising, but this is a mouse model. So we can hope that it won't hold up for humans. Unfortunately, information is starting to come out that these animal models are actually quite nicely predictive. So this is a paper that came out from a pediatric ophthalmologist, Marilyn Miller. She's a lovely woman and happy to talk with any clinicians who have specific questions. But what this study found was looking at some of the earliest recognized infants with congenital Zika syndrome who are now toddlers. So she looked at their vision effects specifically because she's an ophthalmologist. So what she found was that vision impairment was very common, and sometimes this was due to direct retinal damage, other times it was due to optic nerve damage, other times it was due to both. But the important reason why I put this slide up here was that this corroborates mouse model findings perfectly. That's exactly what they saw in the mice. And that's good from a standpoint of validating the animal model. It's bad from the standpoint of what it may mean for humans and what we might see in the future from these babies who are born to infected mothers. So that's not good. Okay, so it's not just, again, the microcephaly and the brain defects that come from Zika's newfound placental tropism. We also see neuropathies in children and adults. The first one that was noticed was Guillain-Barré syndrome. So Zika was confirmed to be in Brazil in May of 2015. What was seen in June of 2015 was a spike in Guillain-Barré cases. And it's actually very nice when you look at the overlays in timing of these two. This is the spike in Zika. This is a spike in Guillain-Barré. They correlate perfectly. And so then what ended up happening was, people went back and they took the Guillain-Barré patients and they looked for evidence of Zika infection, and lo and behold, they almost always found it. And so this presents as an ascending paresis and a tetraparesis. Fortunately, it's treated very much the same way as any standard case of Guillain-Barré syndrome. So you use IV IgG treatment, plasmapheresis, and it actually resolves quite nicely. So this is good because it's a neuropathy, but it's temporary. That is good because there are also some that are not so temporary in children and adults. So Zika has been now confirmed to cause encephalitis and meningoencephalitis. It's also been found to cause encephalomyelitis, transverse myelitis, peripheral facial palsy, all kinds of neurological defects from infection of an adult. And I realize I am belaboring that point, but I'm belaboring it because there's, I think, been a bit of confusion in that people seem to think, oh, the neurological problems with Zika are just the babies who are infected, who are born to the infected mothers. That's not the case. You can have primary neuropathies in adults and older children who are infected. So I put this series of images from a case report from last October. So this is a patient who came in on day 1. And you can see she's got nice, it was a female patient. She's got a nice lesion right here. On day 15, she was clinically normal. So she had no fever. She had she had some ataxia that had resolved. But you can see she's still got remnant lesions in her brain. So what's that going to do to her as she progresses through life? Do not know, but potentially something. And this is one patient, and we have to assume that she represents several others. All right. I just insinuated this, but just to show you directly, there's been now several papers that show direct detection of Zika virus by molecular methods in cerebrospinal fluid. And in many cases, and sorry, these were obviously, because you're taking CSF out of them, these were clinically affected patients. They were clear-cut meningoencephalitis or encephalitis patients. And some of them were co-infected with Zika and chikungunya and dengue, so that's a bad luck situation you got there. Some of them had Zika and chikungunya, but at least one patient in this study and several patients in other studies were infected with Zika alone. You can't read it very well on this slide, but this gives just a quick description of their clinical presentation. All of them were non-negligible. They were all pretty emergent. And the other thing that is critically important that came out of this study and has been confirmed by others, that HIV status was not predictive of developing encephalitis. So one of the things people were hoping for when we started seeing encephalitis was that maybe this is just immunocompromised patients. No, it doesn't seem to be the case. So HIV status wasn't predictive, nor were any other immunodeficiencies, and age was not predictive either. So our one guy down here who had Zika alone and had, I'll read this to you because I don't know if you can read it. He had blurred vision in his left eye. He had temporary blindness. He had weakness. He had loss of muscle strength. He was ataxic. He'd lost sphincter control. He had serious stuff going on. When you look at the paper, he was patient 15. He was 18 years old, otherwise completely healthy. So there was nothing about this guy that would have predisposed him to any kind of neurological invasion, yet he had it. Many of these patients fell in the same category. So that's not reassuring. All right. The other thing that we talk about a lot is ongoing transmission in the United States. How possible is this? How much in Maine do we have to panic about this? One thing, and you'll see this reemphasized in a second, is that we think of this as a vector-borne disease, and obviously it is, but it's also sexually transmitted. That is unique for an arbovirus. This is not something that we see with dengue or West Nile or any of these other guys. So Zika seems to be doing a lot of tricksy things that its cousins don't do, which is unfortunate. This is spread by two different mosquitoes, Aedes aegypti and Aedes albopictus. This is the extremely liberal viewpoint of the range of Aedes aegypti. We used to consider it just here. It's now moving north and west. Yay, climate change. But it's the predominant mosquito in the southeastern United States. Aedes albopictus has a far greater range, and you can see that includes where we are right now. That's at the very, very tip of the range, but the prediction is that range is going to do nothing but expand, which is unfortunate because Aedes albopictus is a far more aggressive mosquito than Aegypti is. It bites all day instead of just biting at dawn and dusk the way Culex mosquitoes do. So if it had to be carried by one, this is not the one you want, but unfortunately, here we are. The other thing that's important to remember, again, that Zika can also be spread by blood transfusion. In fact, so much so that blood donation was halted in Puerto Rico because so many of the donors were infected. Sexual transmission is a far greater problem than we realized. I'll show you why on the next slide. It can be obviously vertically transmitted from women to babies. It's also shed in tears, in urine, in saliva. Basically, any fluidy thing on you can shed Zika if you are infected with it. This paper just came out, so it was EPUB available in July, but it came out to the masses August 3rd, so this is kind of very fresh vintage. So this title sounds kind of dry, but the juicy bits of this paper are in the abstract. Long story short for what they found, so this was a group who does mathematical modeling, and specifically they do outbreak modeling. You can kind of tell there's a lot of math involved by the really jazzy title. But so basically their take-homes were that in the absence of a place where mosquito transmission is happening, so a place like Maine, Zika epidemics can be introduced and sustained as sexually transmitted infections. And then if you are in a place where you have sexual transmission and vector transmission by both Aedes aegypti and Aedes albopictus, such as in Georgia, as Dr. Mokler mentioned, this is a major problem. So in the southeastern United States, for fun factiness, tends to have the highest per capita incidence of sexually transmitted diseases of anywhere in the country anyway. So when you add another one in that can also be vector-borne, that's not good. That's a bad situation. So their take-homes here were that we're currently underestimating the risk of sustained transmission, and that local outbreaks are very much possible without mosquitoes. And so that makes treatment, prevention, and control of Zika all the more critical. It also means that we need to do a lot more understanding of how this virus works and how to prevent it than we currently have, which, considering we learned this was a problem in mid-2015, I'm quite proud of my discipline for how far we've come with this in a short period of time. So that's a perfect segue, is it not, into some basic research for how are we going to come up with some of these good treatment and control strategies. So I want to start by looking at some of the genetic diversity studies that we did in my lab, because they really pointed us to some very nice basic studies we could do. Okay, so quick word about Zika virus. This is a flavivirus. It's got a genome that has 10 different genes in it. They're all stuck right in a row back to back. They're transcribed and translated as one big giant protein, and then they're cleaved into their functional pieces afterwards. That means, from a standpoint of looking at things like, well, what gene is expressed during what phase, we don't have to worry about any of that. So that actually makes this nice and tidy in a way. This genome is 10.5 kilobase pairs, so 10,500 bases. It's teeny, it's easy to sequence. So what we did was we looked at 33 complete genomes, some from the Western Hemisphere, some from Southeast Asia, and some from Africa. And what we did was we looked at nucleotide changes and amino acid changes that were noticeably distinct among these Western Hemisphere strains and the Southeast Asian strains where we think we had the start of these clinical presentations. How were those different from African strains? So what we did was a lot of looking at a lot of sequence we generated what's called a consensus sequence. It means at every single position, we picked the most common base and then the most common amino acid. So just to give you a sense of what this looks like and why I'm so bananas half the time, is I spend a lot of time staring at long strings of letters. So this is 60 of the 10,500 bases that make up this genome. So what you see when you align all these guys is something like this. A lot of letters, and then on the bottom, you see some stars. And then you see some blank spaces. And when you see a blank space, it means that somebody is different in here. And what you can see when you look at this spot in particular is that some of these strains have a C, a cytosine. Some of them have a T, or thymine. So what we did was we looked at, by generating this consensus sequence, we found what is the most common residue at this spot, and then who has a difference, how many of these strains have a difference from the consensus. So what we did was calculated some numbers. So I'm going to walk you through what you're actually looking at here. When we generated our consensus sequence, we went through and we counted how many nucleotide changes we found relative to consensus in any strain. Then what we did was we translated that entire polyprotein, and then we had to look at 1/3 the number of letters, but still a lot of letters. And we did the same thing. We came up with a consensus amino acid sequence, and then we tabulated how many amino acid changes we had in at least one strain. We then normalized that to 100 residues so that we could look at genes that were 2 different sizes and not just count a number, but have a changes per 100 bases or 100 amino acids. And this is what we found. We've got some significance markers up here. So what we did was we counted how many changes we saw per 100 in the capsid protein, in the PRM protein, in the envelope protein, and then we compared that to the number of changes that we saw in the cleavage fragments. So remembering, we have this big genome that's translated as one big piece, and then it's cut into its individual functional proteins. It doesn't cut evenly. It'll cut here and then cut here, and you'll have this little bit in between that gets degraded and thrown away. It doesn't really do anything. So we thought that was a really nice background rate of change because there's no evolution acting on that. So what we found was that you have a certain rate of amino acid change, you have a certain rate of nucleotide change. The gray bars are nucleotide changes, the black bars are amino acid changes. And then it's per each protein labeled on the bottom. What you see here is kind of what you'd predict, that you have more or less 3 times the number of nucleotide changes that you have amino acid changes, because for every 3 nucleotides, you get 1 amino acid. What we found here was that some of these had amino acid changes that were significantly higher than the background rate of genetic drift that we see in those cleavage fragments. A few of them had nucleotide changes that were higher. What that tells me instantly is that there are, first of all, a lot of silent mutations in a lot of these genes. So here we have a change in nucleotide, but we don't have a change in amino acid. So that protein isn't any different, even though you have a genetic change. But what we see in some of these proteins right here, the capsid, the promembrane protein, and the envelope protein, is that you have significantly higher amino acid changes. That tells you that that protein is changing a lot. It's particularly meaningful here because these are the 3 that are on the outside surface of the virus. These are the 3 that are interacting with host cells and interacting with the immune system. So some of this diversity that we saw went beyond random chance. This is another way of saying the same thing. This is a ratio of amino acid to nucleotide changes. The closer you are to 1, the more impactful your genetic changes were. Okay, we also looked at glycosylation patterns. So when you have glycans stuck to the outside of proteins, a lot of times what that does is that it helps a virus interact with a new type of tissue or a new type of host cell. or it helps it hide an epitope. And so if we're looking at a new pathology that we didn't see before, that kind of tells us we're probably looking at a new ability to interact with a different type of cell. And so we wanted in particular to look at these changes. So the way that we can read this extremely busy diagram is as follows. This is what we call markers of O-linked glycosylation. This is N-linked. The way that you read this is that each of these columns here represents one of our strains that we studied. Across the rows, this is one site where we had a potential change in glycosylation, or a potential glycosylation site, I should say. If it's gray, that means every strain was glycosylated. If it's black, that means that strain was not glycosylated. So what we want to see is spots where we have gray here and black here, because that's going to tell us the African strains are either glycosylated or not glycosylated, and the Asian and American strains are the opposite. So this might be a way that they interact differently than the African strains. Does that make sense? All right, so you can study this and try to Rorschach it if you want, but basically the take home here is that there was no smoking gun like that in the O-link glycosylation, but there were in the ends. So we can see here and here, we've got some instances where we've got glycosylation across these strains and none in the African strains. In particular, we zeroed in on this one right here. The reason that we liked it so much is that it was in the envelope protein. So this is on the outside of the surface, and this is the protein that's responsible for binding of Zika virus to host cells. This bit right here, it's actually an asparagine, and it's asparagine number 154 of the envelope protein, if you're interested in that sort of thing. And we liked it for a lot of reasons. One of which was that when we did some structural modeling, we could see that this is a crystal structure diagram of the Zika envelope protein. What we found was that asparagine 154 was right here on this big, flexible, floppy loop. So if we blow that up and flip it around so you can see it a little better, it's not got any kind of tight structure to it. What that means, this is what we call a disordered region, meaning it's very flexible, it can move and bend and twist any way it wants to, without the protein's function being altered. What this means is that this is the sort of thing that usually interacts with host cells. The rest of these beta sheets and alpha helices, these have to be held together in a certain way in order for that protein to do what it needs to do and sit anchored where it needs to sit anchored. If we want to look at something that's different in some strains versus another, a disordered region is exactly where you would expect to find it. And we see this new glycosylation right in that region. So we liked it for that reason too. All right, so now we're going to talk about some cell binding. All right, so to put on a slide the thing I just tried to communicate is when we have new disease states, usually what that means is that you have a pathogen binding a tissue it couldn't bind before. That's what we call tissue tropism. If you have a new tissue tropism, that generally indicates that pathogen is able to bind a new cell in a very unique new way. A new receptor-ligand interaction, for those of you who remember our discussions from week 2, if you are in OMK1. A new lock and a new key. For emerging pathologies, what that means is that if you can bind a new cell, if you have a novel tissue tropism, because you're seeing a clinical presentation that you've not seen before, that usually means that you have a new ability to bind a different type of host cell. So using that logic, we wanted to figure out what was different. Unfortunately, nobody had any idea in the world how Zika virus binds. So we sought to figure that out. So how does it bind? If you look at this, this is a crystal structure of the entire virus itself. The envelope protein are these little pokey red things that come out of the surface and stick out a little bit. That's how it clamps on to a host cell and absorbs and infects. If we look at our envelope protein and our change in glycosylation, what we wanted to do was look at the amino acid sequence in that, around that glycosylation site and say, well, how conserved is it? We know there's this one difference, but are there any others? And it turns out, remember, our stars mean everybody's identical, and blank spaces mean they're not. Colons mean you have a similar amino acid. You can see we've got quite a few differences here. One thing I would like to point out is that everybody at the top here, these are all Asian and American strains, and they are all residue for residue identical. Down here, the African strains are a little more diverse. So we wanted to see, could we take this little stretch of amino acid sequence, if we synthesize it, and we suspect that the way that Zika virus binds is contained within this little bit of sequence, if we just make a little synthetic fragment of that, that should bind host cells. And we know it's not part of a secondary or tertiary structure because it's part of this big, flexible, linear loop. That was why looking at the crystal structure of that protein was so informative to us. It told us that we could probably do a study like that, where we take this little synthetic fragment and feel reasonably confident that it represents virus binding. So we did that. We generated some synthetic peptides, and we put a FITSI label on it, so it's a big yellow floor, or a green floor, I should say. And we synthesized a few of these because they're expensive, so we didn't want to synthesize all of them. So the first thing we did was we made this strain right here, which is from Puerto Rico in 2015, and we put an N-acetylglucosamine onto asparagine 154. So that's the sugar that's predicted to be stuck on that. So we synthesized that and stuck one on. The only thing different about this from how it naturally occurs is that this has a floor on the end. So then we also made the same sequence, Puerto Rico 2015, but did not put a NAG tag on it, so it's just naked. And then we synthesized the type strain from Uganda, and then we synthesized another strain from Nigeria that was from the 60s that had some deletions in them that we thought that might be interesting. It might help narrow down what residues are important. As controls, what we did was we took this sequence of amino acids and we scrambled it up. So same chemical content, but just in a different order. And then we did the same exact sequence, but we put a NAG tag on this one as well. So these we expect to be negative controls. All right. What we did first was we wanted to look at some neurons to see if these guys bound. So the help of Derek Molliver's lab and done by Diana Good and Ramaz, whose last name I can't say, they collected some primary dorsal root ganglion neurons from mice, cultured them in chambered slides, and we allowed our peptides to bind. We washed them away by just by rinsing with HBSS, and then we visualized where the cells were by staining them with DAPI, so that's a blue fluor. What we found was that our naked Puerto Rican strain, you could see that you had some green staining right around where those nuclei were, so we thought, it's interesting. With the Ugandan strain, we saw the same thing. You have nuclei here, and you have some nice staining right around that. Saw the same with the Nigerian strain. When we looked at the scrambled control, you can see some green gunk in the background, but none of it is really clustering around nuclei the way they are in the other cells. So we found that to be fairly interesting. We wanted to then quantify that. So what we did was we developed an assay because there was no roadmap for this. We were, frankly, making things up as we went along. We grew cell layers. We used MDCKs and Vero cells, and we put our tagged peptides on them. And then what we did was we simply measured fluorescence. And when we did that, this is what we saw. So these data haven't been normalized yet. I apologize because I ran out of time this morning. But one of the things that we found was that we saw some differences. When we do a straight ANOVA on these guys, you see a significant difference between the naked Puerto Rican strain and its scrambled control. You see a difference between the Ugandan strain and the scrambled control. And you also, interestingly, you don't see a difference between the NAG-tagged strains. I think in some ways that's because NAG binds onto a whole bunch of host cellular proteins, so that might be having that peptide stick regardless of what order it's in. But that being said, we still have to normalize these, so don't take them to the bank right now. When we looked using confocal microscopy on Vero cells, we saw some really cool stuff. So this is looking just at the FITC. So these are the peptides from the Ugandan strain, the unnagged Puerto Rican strain, and the nag-tagged Puerto Rican strain. This is the DAPI, where you can see where the cells are. And then this is the merged images, where you can see that you have this very nice co-localization. So those peptides are binding on to those cells very nicely. And this is the control images, where you can see you've got some junk in the background and some cells, but not much co-localization. Sorry, I'm speeding myself up because I'm behind. So we found that peptides bind. We wanted to see, is this the binding mechanism, though? And in order to do that, we tried another type of experiment, where we grew a Vero cell monolayer, and we wanted to put peptides on them to make sure they stuck on to whatever the lock was on those host cells. And then our prediction was, if we tried to infect with live Zika virus, the viruses wouldn't be able to infect anymore because their binding sites have peptides stuck on them. And so they can no longer absorb. That was our hypothesis. So we were looking for a reduction in infection rate when we pre-treated with peptide. And that's what we found. So the exciting thing here was that when we, just to give you the point in a nutshell, is when we looked at the Puerto Rican strain, the unglycosylated peptide from the Puerto Rican strain, it inhibited the Ugandan strain, so the African strain. When we looked at the Puerto Rican strain that was nag-tagged, it inhibited the Puerto Rican strain. So you have the peptide with the glycan inhibiting the Western Hemisphere strain, and the peptide with no glycan inhibiting the African strain. That was more or less what we predicted. It gives you an idea that that change in glycosylation is key to the binding differences that we see in these two families of strains. So we were really excited about that. As per usual, I get excited, and then 10 minutes later, I start deciding there's all kinds of other things that we need to do, which is why I make Courtney bananas. But anyways, we also had some interesting data when we looked at MDCK cells. So we used the same exact experimental approach as with Vero cells, but we found something very different. What we found here was that when we looked at the binding of the Ugandan peptide relative to scrambled control, this was almost significantly different, but not quite, which is really irritating. But one of the things that we did find was that the peptide with no glycan on it was significantly different from its scrambled control in its ability to bind MDCK cells. Big difference was that we found when we nag-tagged it and then looked at the scrambled control, they were not only not different from each other, they were wildly different from the unglycosylated ones. They were no different than the PBS background, which to us, we looked at that and said, geez, it's almost like when you put the nag on it, it completely diminishes the ability of that motif to bind this type of cell. And so we said, but the virus still infects that type of cell, so what's going on here? So we said, it must be using another thing. What is it doing? So we said, is there another way of binding? So then we looked at some clues to help us answer that question. The first was that a paper had come out several months ago saying Zika virus is reported to bind a protein called AXL on host cells. There are a whole bunch of viruses that can bind this protein, and they do so by incorporating a phospholipid called phosphatidylserine into their envelopes. This is a non-specific viral binding mechanism that is used by a ton of viruses. That's clinically important in a second, so hold that thought. we had a hypothesis that if we could bind phosphatidylserine on a viral cell surface, that would inhibit viral infection as well. And fortunately, there's a protein that does that beautifully, called an Nexin V or an Nexin 5. I've heard it said both ways. I'm just going to say 5, because I like Roman things, I guess. Anyway, the point being, so we set up an experiment where we did this. We grew a viro cell, a viro monolayer, and we took infectious Zika virus and we pre-incubated it with a Nexin 5, assuming that this would happen. The Nexin 5 is going to bind onto the phosphatidylserine and inhibit viral absorption and drop the rate of infection. And guess what we found? This has all the hallmarks of an 11 p.m. modification, because that's when I finished doing these stats and I danced around my kitchen a bunch. This tells you how hot off the presses these data are. So when we looked at an Nexin 5 and we absorbed it to the African strain of Zika virus, we had no difference in infection. When we pre-incubated the Puerto Rican 2015 strain with a Nexin 5, we had a significant difference in infection. So that tells you Western Hemisphere strains seem to be capable of infecting using phosphatidylserine, and African strains do not. So this now has two different ways of interacting with host cells. And that's going to give you new tissue tropisms and new clinical presentations. In particular, I was excited to see this phosphatidylserine work out because it's a druggable target, and it also explains why these strains were more aggressive and more invasive than these, because this ability gives this virus that attachment ability gives that virus the capacity to bind many, many, many types of tissues, and clinically that's what we see. So it made a lot of sense, really hung things together. So future directions. This has some CME and some basic research in it. I want to talk about future directions in the context of clinical implications and applications first. First thing we saw was that unglycosylated or African-type peptides blocked the African strain. Glycosylated or Western Hemisphere type peptides blocked the Caribbean strain that we used. And this change was biologically relevant. So it stopped infection, or I should say it didn't stop it, it reduced infection. So if peptides block viral absorption, do they have therapeutic potential? That's interesting. That's kind of neat. We also saw that blocking phosphatidylserine interrupted viral absorption. Does that have therapeutic potential? And that's kind of cool because that goes beyond Zika. That's for a whole bunch of different envelope viruses. And it turns out these are my peptides that we made and just fool around with in our lab as proof of concept. They're not, you couldn't inject them into a patient. But this, it turns out, that is a great target for drug repurposing. I want to put this up first because this is a paper that showed a whole bunch of TIM family proteins, one of which is AXL, seem to be targeted by virus-associated phosphatidylserine, including flaviviruses, which is what we experimentally demonstrated with Zika as well. No one has yet demonstrated it with Zika until Courtney and Dr. Vaughn and I. Okay, not I, they did. I just sat in my office and typed things. I want to also bring up this paper where this group suggested that blocking phosphatidylserine might be a nice antiviral and a nice broad spectrum antiviral, of which there really aren't any right now. So what they did was they used an immunologic drug that was in clinical trials called bavituximab. This is a monoclonal antibody that blocks phosphatidylserine. They experimentally infected guinea pigs with something called Lassa fever virus, and they gave these guinea pigs IV injections of bavituximab, and they got better. They completely and utterly got better. And Lassa, we haven't learned this yet in OMK1, but any second years hopefully remember, this is a hemorrhagic fever virus. It's got about a 40% mortality rate. So if you can cure it, that's pretty darn impressive. So I mentioned this was in clinical trials. Turns out it was FDA fast-tracked 2 years ago. In its clinical trials, it was developed as a cancer drug, but doesn't matter. Its clinical trials just finished a couple months ago. So it is now in FDA review. So this is the kind of thing that's a perfect target for drug repurposing for not only Zika infection if you had a pregnant patient come in who had been exposed, but it's also an excellent target for drug repurposing for any enveloped virus that might emerge in the future. Something like, I don't know, Ebola or Lassa or something like that. If you have a patient wander in who's critical, it could be worth a go. Okay, so it gave us, our work gave us another key clinical question here, which was, does adding NAG to asparagine 154 in the envelope protein give Zika virus tropism for the brain and the placenta? So that's our thing that we are going to figure out next. And we're going to do that with in vitro studies using a pregnant mouse model. So we have those scheduled for September, which is like 10 days from now. So wow. So we have to get on impregnating some mice in the not too distant future. So what we're going to do is use our peptides to see if we can traffic or observe trafficking to the brain and the placenta or any other organs. All right, so finally, the future clinical implications here would be that understanding binding mechanisms really helps us define tissue tropism. It's a clear-cut example of basic research and laboratory bench research that has a direct clinical correlation because tissue tropism dictates clinical presentation. What cells you bind tells you what part of a human gets sick. And then finally, this also helps with prediction of future emerging diseases because it can help us alert clinicians to potential presentations. If we figure out a binding motif, I can put that in a database. Any new crazy virus that pops up that somebody sequences, what they are going to do is search that database for protein functional motifs. If our functional motif pops up and it says, this binds this way, people can then know, oh gosh, that's going to have placental tropism. Watch out for pregnant patients. That can happen in the span of 5 hours. That's pretty impressive. To go from a sick patient to sequencing to knowing what it can do. That's pretty impressive. I'm jazzed about that. So in summary, Zika is, from the CME side of things, Zika still continues to be a rapidly developing situation. The majority of patients who come in are subclinical, and of those that are not, most of them recover fully, but neurological presentations do occur in children and adults. Congenital Zika syndrome is going to pose a very, very significant long-term public health and public health infrastructure problem in all countries in the Western Hemisphere. Outbreaks in the southeast and eastern US are highly possible, if not probable. This problem is not going anywhere. The Western Hemisphere strains are derived from one emergent clone, one patient, one time, seeds a whole hemisphere. And then these are very genetically distinct from the African strains. And then finally, we sorted out the binding mechanism of Zika virus here at UNE. Go Nor'easters. And we found that a NAG modification of the envelope protein at asparagine 154, which sounds nerdy, has a whole bunch of clinical implications. So with that, I want to acknowledge a whole bunch of people. The first person is Courtney Reeder, who does all of this work, which is incredible, and she also deals with me on a daily basis, which I'm sure no one envies. We also I have to very much thank Dr. Jim Vaughn, who's done all of these infection inhibition studies in his lab. So don't come to the 4th floor because there's Zika all over it. But that's something that we've worked together very tightly on. I want to thank John Reeder, who had part of this as his Morgane Fellowship project. I have to thank Pete Caradonna and Ivy Bergquist, who are of UNE's core facilities, who Pete taught us how to do all of this imaging that we did not know how to do. And Ivy's helped us very much in planning these upcoming in vivo experiments. I do thank the Molliver lab, so Eric and Diana and Ramaz, who extracted DRGs for us so we could do that first round of imaging. And as we discussed in class this morning, some more imaging in a few weeks. We figured out what we're going to do with the male mice that are no longer needed once they impregnate the female mice for the Mo study. And then I also want to thank Dr. Tamara King and Sebastian Sanjas for helping plan these studies. And it's more of like a future thank you because they're running these studies. So I have no idea if I have any moments for questions, but I'd be happy to take any if I do. Oh, sorry, one more thing. I want to point out before Dr. King asks her question. All of this was IDC funded, and funded in some cases by in generosity from my chairman, who advocated and lobbied for me to get some bridge funding, because our NIH grants are pending because this is so new and needed to be done so rapidly that often you can't wait for 9 months of a funding cycle to come through. So I figure we discovered a lot, shoestringing it and throwing things together at the last minute. So, thank you.
I actually was going to say you do have time for 1 or 2 questions.
And then I took more of it up. So.
Now maybe 1. But I will save my questions because I see her all the time. Does anyone have any questions? Please, feel free.
Yeah, if I'm ever, sure.
Hello? Okay. So it's wonderful. That's great work, really, Megan. I'm really impressed. I had no idea what you've been doing. It's really great and really applicable, and that's really the exact thing that we need between basic science and clinicians. So my question is, so you identified the part of the glycosylation, part of the virus. Mm-hmm. So do we expect with that consistent mutations that happen that could change and something else would come up, and then we are confronted with resistant strains and we're unable to, yes?
That's an outstanding question. So flaviviruses have an incredibly high rate of diversification. So they're not quite at flu or HIV level, but they're next tier down. So they do change a lot. However, one of the things that I thought was kind of nifty from that ratio of nucleotide to amino acid changes, one of the things you can suss out of that is that only certain proteins can tolerate above average levels of diversification. So you might see nucleotide, but you often don't see protein. However, that said, the envelope and the capsid and the promembrane proteins are the one that you worry about most changing. And I agree with you. And what it comes down to, I think, is that if we see changes that virus finds favorable, we might be looking at something different a few years from now. And so I will say it doesn't seem to have an elevated rate of diversification relative to any other flavivirus, but that being said, they are pretty diversified. They diversify a lot. We see that with, if you look at how many different subtleties there are between the 4 dengue serotypes, the fact that there are 4 dengue serotypes would be exhibit A. The clinical diversity you see in yellow fever cases, in West Nile cases, I think all of those, you're looking at something exactly like what you're saying, that you have these minor diversifications and they have clinical relevance. A good question, thank you. Dr. Good.
So phosphatidylserine is usually expressed on cells that are activated or prior to going apoptosis, right? Mm-hmm. So in your culture system, did you notice, like, is there some, like, how are these cells activated? Because they're not probably expressing PS on the surface.
Right, so the cells aren't, the viruses are. So the PS is on the viral envelope. Gotcha. I know, it took me a minute too when I was reading. I know it's really counterintuitive, but it's on the viral envelope, and they're using that as a receptor. Dr. Spicer. Thank you.
That was a great talk. The anti-phosphatyrosine antibody, what type of not off target, it would be phosphatidylserine, but are there side effects, as Diana said, that it activated cells have it, so would they have problems?
Right, so does it serve as like a cross-linker to cells that are expressing it in the body? That's a really good question. To be honest with you, I don't know the answer. The hint that I think we can take out of some of those cancer publications where this company that developed it called Peregrine, starts with a P. It made me think of Falcon, so I'm going to say Peregrine. That they published is that their side effect profile was lower than standard chemo. So, yeah, that's good, but it might not be great. It may not mean that there's nothing, but it may mean that it's not quite so toxic as standard chemo, because again, this was developed initially as a cancer drug. And just some clever virologists in 2009 said, hey, wait a minute, I think this could also be an antiviral. But in terms of using it for in terms of its side effects, it's a legitimate concern, but I can say it's not lethal, and it's less than standard chemo. Oh, sorry, Dr. Davidoff.
No, that was a great talk. And what I love about talks like this is I have 100 questions, and I had to cross them all out because you answered almost all of them.
Huzzah!
And that's not easy for me to say.
I take that as a compliment.
I do have some very naive questions because viruses is not my thing. And so if...
They weren't my thing until a couple of years ago either. It's weird.
If the phosphatidylserine blocking, is that effective? Could you anticipate taking a non-Zika virus and inhibiting or reducing infection as well? I mean,
yes.
Is that predictable? Did I miss you saying that, or is it?
Yeah, so I said it in clinical parlance. So this is something that you could use as a broad spectrum antiviral, and that's exactly what I should have said in basic science parlance, which is that could work theoretically on any virus that uses PS as a receptor.
So my question was, is that the next experiment?
Well, the next experiment is the pregnant mice, and then we got to get writing. And then perhaps after that will be aim 3 in the R01 that Tamara and I are writing, that she doesn't know about yet.
And so just one little knucklehead question. You near the end, and I think it was your, I can't remember if it was your peptide binding experiment or the phosphatidyl or the Annex and 5 experiment, but you showed that the African strain had a whopping variability of the rate of infection.
Yes.
And so that wasn't significant.
But yes,
the
it had a very
Western
high,
strain was. So there, yeah, that
yes.
one.
This had a terrible standard deviation, as you could imagine. Yes, so that's something that Dr. Vaughn and I spoke about because we wanted to fix it and have it be prettier.
It's called non-parametric statistics.
Sounds like a Dr. Williams thing. No, but so basically what we did was we talked about doing this, again. And so he did that again. And I don't have the numbers yet. Sorry. I didn't label this axis, but this is a TCID 50 score. So those take probably about 12 days to get the end result of. So we're on like day 8. So
maybe we could talk about it later.
Yeah. I know I can do it mathematically, but we just figured we could do it biologically and be thorough because we're both anal about that sort of thing.
All right, so unfortunately I have to end this because they have to prep for the afternoon
Yeah,
session.
exactly. I have to teach in 15 minutes. In case you people haven't heard me talk enough today.
But I want to thank everyone and thanks for the excellent questions. Thank you, Megan.